All right, so treating cancer and thinking about evolution. Again, this is a major theme in that book that I described at the beginning of this lecture. Now, this is a figure that describes how cancer evolves. Okay, we're talking evolution in a patient even before there's intervention or a chemotherapy applied. And so you want to look at this figure, and on the far left, there's these green circles. Those represent normal cells. And then at some point, there was a mutation whereby a normal cell turned into a cancerous cell. And that means it's going to have those traits that I described at the beginning of the lecture, whereby it's going to lose regulation and differentiation, but also proliferation, too much division. It's going to potentially monopolize resources, etc. And so once you have that first cancerous cell and it is reproducing, every time it reproduces, remember, there's an opportunity for more mutations to occur. And so this string of this letters and these stars here represent names for mutations in the genes. And so this figure is showing that the founding tumor is in this gray, but then eventually there becomes cancer cells that are different than the original ones. And so in the figure, they've been designated these different colors, including this purple and yellow, etc. Different, it's called clusters in this figure. So each of the colors represents a lineage of cancer cells, and they'll have different mutations. So anyways, at some point, if this patient is getting treatment, they may be given a chemotherapy. And so what happens here in this particular figure, chemotherapy is applied. And then you see the, I want to make sure you guys can see my pointer. I hope you guys can see it. Let me see. What you can see is that the colored bubbles shrink in width, and that's supposed to represent that the cancer cells, there's less cancer cells after chemotherapy than there is before chemotherapy. But you'll notice that the cancer cells don't disappear altogether. Now the purple bubble stops, and so we could say that those purple cancer cells went extinct, but the yellow and orange varieties continue to live. And so after the chemotherapy period, the yellow and the orange cells went back to dividing, and eventually their population size grows, this bar gets wider and wider, and then a new form comes, and it's this red. And so after chemotherapy, there's a reduction in the number of cells, cancer cells, but some survived, and then they rebounded in their population size. And so if you're going to look at cancer over an evolutionary tree, what you can see is that at the base of the tree here, there's the normal cells. At some point, there is a new lineage that's formed because there's mutations that accumulated that allowed for a cell to become cancerous in this gray. But then even once there's the gray cells, there's other mutations that lead to new clusters. And so again, similar to bacteria or viruses, an analogous here would be strains. So they're all sourced from the same original cancer, but they're different enough that scientists call them different clusters, just like you might call different bacteria, you might call them strains. And so this is what an evolutionary tree will look like for cancer. And you can see that in this example, the purple line ends. And so in this case, that cluster is extinct, and current time here is on the far right. And so what's happening is a natural selection scenario whereby you have an initial tumor. Here's another figure. You have an initial tumor on the left of this figure, whereby there are some cells that are chemotherapy sensitive, meaning that chemotherapy would kill them. And then there's some that are chemotherapy resistant, meaning that the chemotherapy would not kill them. And remember that this would be very drug specific because every drug has different modes of action. And so a conventional cancer therapy or chemotherapy, chemotherapy just means to use drugs as a therapy, would be to give a patient a maximum tolerated dose. So how much of the drug can you give the patient without making the patient too sick? Because there tends to be lots of side effects with chemotherapy drugs because they're designed to kill human cells, right? It's hard to have them target just cancer cells. And so some healthy cells often get killed as well. But what can happen is when you apply that maximum dose, a lot of the cancer cells are going to die, but if there happens to be a variation that can resist the chemotherapy, it will survive. And so when that resistant cell is the only survivor, then it's going to replicate, and eventually you'll have a tumor here at the right of the figure that is all chemotherapy resistant. And so you cannot use that drug to try to kill off this tumor because all of these cells are resistant. And I hope that you can see that this natural selection scenario is very similar to the other one that I described as a treadmill, not only with pesticides used in agriculture, but also with antibiotics and antiviral drugs. So the problem with using high dose chemicals in these different scenarios is that if there's a resistant individual in the population that you're trying to kill off, natural selection allows us to very accurately predict that those resistant members will survive, and then the next time that you use that chemical, it won't be useful anymore, and then you just need a new chemical, okay? So it's very difficult to treat cancer with chemotherapies for this reason.